Welcome to our microlearning channel. In today's video, we will share on bioinformatics and biotechnology databases. We first start by showing how biological data has exploded over time and why NCBI became essential. Sequencing costs plunged and data volumes surged far beyond historical research capacity, creating a massive bottleneck. On the right, you can see how isolated lab silos were unified into an NCBI hub that stores, organizes, and democratizes molecular data so scientists worldwide can share and analyze it efficiently. Here we highlight the Entree Query system as the central hub that links different data types into one searchable ecosystem. The diagram shows literature, genomes, proteins, and clinical data as spokes that converge on a global query engine, making cross-references and integrated searches possible so researchers can move smoothly from a paper to a sequence or from a protein to clinical evidence. This funnel graphic summarizes how PubMed and PubMed Central turn millions of citations into accessible biological knowledge, with full-text articles feeding integrated data resources. The visual mock-up shows how text mining can jump from an abstract to a DNA sequence, illustrating that literature isn't just reading material, but a bridge to genomic data and interactive knowledge gateways for discovery. Think of the data pipeline as a ledger that transforms raw sequencing reads into polished reference genomes. The base layer, the sequence read archive, holds huge raw data sets, GenBank collects community-submitted sequences, and RefSec sits at the top as a highly curated, non-redundant reference set that provides a reliable baseline for biological and medical research. This slide illustrates BLAST as the analytical engine that takes a query sequence and searches the NCBI database for matches, highlighting aligned regions in the middle. By rapidly identifying similar sequences, BLAST helps infer protein function, reveal evolutionary relationships, and flag potentially pathogenic mutations, turning a string of letters into actionable biological insights. Here we trace how basic genomic data moves into clinical impact. Raw data are aggregated and processed by NCBI services like ClinVar and DBSNP, then standardized and interpreted to become usable clinical information. The flowchart emphasizes that this standardized genetic data underpins real-world patient applications and ultimately supports life-saving precision medicine decisions.